Hi, biology peeps. Today we're going to talk about enzymes. If you remember from our biomolecules presentation the last time, enzymes are special types of proteins that have a job in our cells, and that is to speed up the chemical reactions inside the cell. And we're going to talk more about that today. Proteins are made of amino acids. Take a look at your notes. The first one asks you, what monomer are proteins made of? The answer to that question is amino acids. Proteins are made of amino acids. We've been through this now 3 times. Hopefully we understand what a monomer is and that the monomer of proteins is amino acids, okay? These diagrams that you're seeing on this page, this one down here, and these up here, these are diagrams that you're going to find a lot on the internet. You also are going to see this diagram right here on a quiz that you'll be taking later this week. Amino acids are strung together with peptide bonds, and they look kind of like beads on a chain at their primary level. So if you take a look at this drawing down here, we've got amino acid, peptide bond, amino acid, peptide bond, amino acid, peptide bond, amino acid, peptide bond, and on and on and on and on and on. This big long chain of a polypeptide. Small proteins are called polypeptides. Take a look at your notes. What is another name for proteins? Another name for proteins is polypeptides. Poly means many, and peptide is the type of bond that is between the amino acids. Again, this is the 3rd time you've seen this, so we should be getting it by now. And here's a closer picture of the amino acids, all kind of linked in a little chain area, and I'm kind of putting little red dots next to those. In between each one of those amino acids is a peptide bond, which is why proteins are also called polypeptides, many peptides, okay? Enzymes are one of the many kinds of proteins that are made by cells. Take a look at your notes. Enzymes are a form of which biomolecule? This is the 3rd one on your notes. Enzymes are proteins. I want you to listen to my words. Enzymes are proteins. Hey friends, enzymes are proteins. I'm going to say that about 100 times over the course of the next few days. What are enzymes? Enzymes are proteins. Enzymes are proteins. Hey guys, enzymes are proteins. The reason I'm saying this over and over is because this is a question that is frequently, almost every year, asked on the STAR exam, which is why I just put a star by it. Because remember, when we have something that we know is going to be asked on the STAR, we put a star by that. Enzymes are found in every cell of your body, and their job is to speed up chemical reactions inside the cell. The next one on your notes asks, what do enzymes do? The answer is they speed up chemical reactions inside the cell. Write it down. Okay, so we need to understand what that means by saying speeding up chemical reactions. Yeah, I know there's a graph, and graphs are scary, but you're going to see a graph like this probably on the STAR. They like to give it. So every chemical reaction in a cell requires some type of energy to get it started. This is known as the activation energy. Activation means to start. I usually give an analogy with your cell phone. When you get a new cell phone or a new cell phone number with a carrier, you have to do something to that phone before you can use it. What you have to do is you have to activate it. You have to call into a number to activate it. Activation means to start something. So activation energy, here's it is right over here, is the amount of energy needed to start a chemical reaction. Take a look at your notes. Define activation energy. That's E sub A. It is the energy required to get it started. What is it? It's a chemical reaction. Okay, so I'm going to write what you need to write on your notes right here. Activation energy is the amount of energy needed to start a chemical reaction. Okay, the amount of energy needed to start a chemical reaction. That is activation energy, or E sub A. So enzymes speed up chemical reactions by lowering the amount of activation energy needed to start a chemical reaction. Take a look at your notes. How are enzymes able to speed up chemical reactions? By lowering the amount of activation energy needed to start the chemical reaction. Before you try to skip over to the next thing, listen to my words. This graph right here, I'm putting a star by it. Why? Because it shows up on the test almost every year. So we need to look at this graph. Along the bottom, right, is time. This is reaction coordinate, but this is actually meaning time as the reaction goes forward. And along this side is energy. So the amount of energy goes from 0 to whatever. So energy is going up this way. And the amount of time is proceeding this way. Okay, so this is our X and this is our Y. When you look at this graph right here, the reaction is starting right over here. And as it proceeds, you can see that there's a time where we've put the chemicals together, but they're not doing anything. That's what I just showed you in red here. Now, without an enzyme, the amount of energy needed in order for that chemical reaction to occur is all the way up here. So you can see how steep this curve is, okay? And then this is the reaction beginning, this is the peak of the reaction, and then this is the chemical reaction's kind of finishing up. And then down here is where the chemical reaction is kind of being done and we're getting our products. So over here we have our things that we're going into, reactants. And over here at the end of our reaction, we have our products, okay? When we add an enzyme, remember enzymes speed up chemical reactions by lowering the activation energy. When we add an enzyme, we still have this little part in the beginning where nothing's happening because the reactants haven't begun to react yet. When we add the enzyme, the amount of energy that we need to start that reaction is much lower, right? So the reaction can go faster because the curve doesn't have to get quite so high. And then, of course, we still have the back end of that coming down here as the reaction kind of finishes up and we get the products. So this is the big takeaway right here. Check this out. Look at how much activation energy is needed without adding an enzyme versus the small amount of energy needed after we add the enzyme. So we don't need as much energy to start the reaction, which speeds up the reaction. Enzymes speed up chemical reactions without being changed in the process. Look at your notes. How are enzymes affected by the reactions they direct? Enzymes speed up chemical reactions without being changed in the process. Write that entire sentence on that line. Enzymes speed up chemical reactions without being changed in the process. This means that an enzyme can be used over and over and over again by a cell. The diagram that you see down here is on your quiz. Did you hear my words? It's on your quiz. This is kind of a famous diagram, and we're going to again take it piece by piece. So we haven't talked really about the parts of this yet, and I'm just going to mention it because we'll get into it. There's some on your notes that we're going to be drawing on. But we're going to start. This is our before picture, okay? This little thing that kind of looks like half of a broken egg down here is the enzyme down here, all right? This is the enzyme, this thing that kind of looks like the broken egg with all the little peaks and valleys, the one that I'm outlining in red. The substrate is this little part up here that's got the unique shape, right, that fits right inside the peaks and valleys of the enzyme. The little part where they fit together is called the active site. And we're going to go through this a lot more in the next couple of slides, but we're doing it now because I want you to take a look at this diagram. As the substrate comes down into the enzyme, notice that they fit together perfectly. We call that the lock and key hypothesis, okay? Because we've got enzymes and substrates that are designed to fit together like a lock and a key. They're unique to one another. We call that the enzyme substrate complex because it's an enzyme paired with a substrate together. Okay? Now, notice what color our substrate is in this picture and in this picture. It's green. That means it hasn't changed chemically yet. It's the same as it was when we started. But once the enzyme does its work on the substrate, look at the color change, right? Okay, so here's our enzyme down here. Notice the enzyme has not changed shape, but our substrate is now different, right? Our substrate has changed from our green bound together to our blue and our orange that are now broken apart. So the enzyme has changed the substrate in this chemical reaction, but the enzyme itself has not been changed. If you take a look at the enzyme in each one of these pictures, there is no change in the enzyme. So the enzyme can go through the chemical process again and unlock or break apart those products again and again and again and again. Okay, before we move on to this slide, take a look at your notes. The one that we need to fill in here says, how many times can a single enzyme be used? And that's what we just talked about. A single enzyme can be used over and over. And I'm going to write that right here. Used over and over. A single enzyme can be used many, many times because remember, the enzyme is not changed during the chemical reaction. All right, so let's move on and talk about this slide right here. Any substance that speeds up a chemical reaction without being permanently changed is known as a catalyst. Look at your notes. Define catalyst. Here it is. Any substance that speeds up a chemical reaction without being permanently changed. There is your answer. Write that entire thing on the line. A catalyst is any substance that speeds up a chemical reaction without being permanently changed. So enzymes are catalysts. Enzymes are catalysts. There are many different catalysts that are used by chemists in the lab, but cells always use enzymes as their catalysts. Look at your notes. What kind of catalysts do cells always use? They always use enzymes. Oh, and by the way, this graph right here is on your quiz. So again, I'm trying to point out to you things that you're going to see again on the test and the quiz so that you can make a note of it in your brain. Okay, so the reactants or the raw materials that enzymes start with are called the enzyme's substrate. Here is the substrate, and we went through this on the previous slide, but we're going to do it again. In this particular graph, you can see that the substrate's shaped a little bit differently, but it's still shaped exactly perfectly to fit inside its unique enzyme. So take a look at your notes. What do we call the reactant that attaches to an enzyme? We call it the substrate. Write it down. And again, in this particular slide, here is our substrate. it has a unique shape that fits perfectly within its enzyme. Because remember, we call this the lock and key hypothesis. Enzymes and substrates fit together like lock and key. So as the reaction proceeds, and remember the active site is this part right here where they fit together. Right here, I'm making it real big and dark right here. Okay, so once they're together, we call it the enzyme substrate complex because they're together and it's a complex. And then as the reaction proceeds, the products are what we get at the end of the chemical reaction. Notice that they've been broken apart. So the substrate's attached to the enzyme at the active site. Okay, so look at your notes right there. We are labeling the diagram that is on your notes right now on this slide. You've got a set of words. You've got enzyme, enzyme substrate complex, substrate, products, and active site. Look at the thing that's on your screen right now. So here we go. So where's your enzyme? Your enzyme is right here. Label it on your diagram. And the enzyme, again, you can see that it remains unchanged in all three of those pictures there. Okay, where's the enzyme substrate complex? Label it. It's when the enzyme and the substrate are locked together. That's the middle diagram. Okay, substrate. Here is your substrate. Okay, it is the reactant that fits inside the enzyme. Products. Where are the products? Label them right here. There they are. Label them on your notes, on your diagram. Active site. Label the active site right here on your diagram, on your notes. There's your active site. It is the place on the enzyme where the substrate and the enzyme click together, where they actually attach. All right, there's another one on your notes that has to do with this slide. It says the substrate always attaches to what part of the enzyme? The answer there is the active site. That's the one, the blank that's underneath the picture on your notes. The substrate always attaches to the active site. All right, got it? Write it down. Moving on. Because the active site is designed to attach to a very specific substrate, cells must have a different enzyme for every chemical reaction. Again, I told you that every enzyme is unique, and you're probably going to see that word, U-N-I-Q-U-E. Enzymes are unique to their substrate, just like a key is unique to a lock. Every enzyme unlocks a very specific and unique substrate. Okay, so take a look at your notes. Why does a cell need a different enzyme for every reaction? Here's the answer. Because the active site is designed to attach to a very specific substrate. They fit together like a lock and key. This is now the third time I've said this. Just any key does not fit in any lock. The same is true with enzymes. Because the active site is designed to attach only to a specific substrate, cells must have a different enzyme for every chemical reaction. Write it on your notes. Okay, so because of this, even the smallest cells have over 1,000 different enzymes inside of them. So the last one on the front page of your notes says, how many different enzymes are in even the smallest cells? Here's your answer. Over 1,000. There are lots of enzymes inside even the tiniest of cells. Okay. Okay, so watching the short video on enzymes, we're going to watch that together in class. There's no way for me to embed it in this particular PowerPoint. I'm going to give you a link to it. But take a look at the back of your notes. Flip to the back of your notes. Okay. This little cartoon of a superhero is a way that we help you remember what enzymes do. All right. Does this superhero look like he's going fast or slow? Okay. I hope that you can see that he's got these little speed lines coming out behind him, right? That means that he's going fast. We want you to think of enzymes right here as speeding up chemical reactions. Enzymes make things go faster. So the enzyme is the crime-fighting catalyst, making your metabolism go faster, one chemical reaction at a time. So on the back of your notes, what is the enzyme's nickname? There it is, right there. The crime-fighting catalyst. Enzymes are catalysts. Catalysts are anything that speeds up a chemical reaction. So what's his superpower? Here it is right here. Speeding up your metabolism, one chemical reaction at a time. So that's the last one on your notes. Speeding up your metabolism, one chemical reaction at a time. Now I'm going to add one more thing to this slide, and you're going to add it underneath, okay? I'm going to do it on the next slide. So I'm going to add a slide, and you guys are going to add something to your notes. Okay, so the next thing that I want you to write down is what hurts the enzyme, because we're keeping with this same superhero theme, right? You guys know that Superman has something that hurts him, right? Superman, the thing that can kill Superman or injure him is kryptonite. Well, the last time we got together, we talked about something that denatures enzymes. So what hurts the enzyme, our crime-fighting catalyst? Heat. He cannot be exposed to heat, okay? And then we're going to write there, enzymes get denatured. And we learned that word last time, denatured, when exposed to high temperature. I'm writing it down, and you should be too. Okay, remember the word denatured means that the enzymes are broken down by the heat and they can no longer function. So if we're thinking of an enzyme as a superhero catalyst that speeds up your metabolism, right? Then the thing that hurts this superhero, the catalyst, is heat, because enzymes get denatured when they're exposed to high temperature. So that's like his quote unquote kryptonite. Hope that makes sense. Add that to your notes. Beneath that thing about his superpower, you're going to add this, what I've just written on this slide. Okay, thanks guys.